Welcome everyone, and welcome to our weekly platform trial webinar updates. We're waiting just a few seconds for people to join us from the waiting room. I see that the numbers are going up, so thank you so much for joining us as we meet and we gather every week. It's really always a pleasure and great appointment for us to meet with all of you, old friends and new friends. Thank you for joining. And if you can go to the next slide. Tonight, it's an absolute pleasure for me to introduce my close colleague, Dr. James Barry, also at the Mass General Hospital, who's the co-lead of the regimen C being tested in the platform trial. But for tonight, he joins us especially because of his amazing expertise in biomarkers. I know we receive a lot of questions about biomarkers on these webinars, and so we really wanted to bring in the expert to help us sort of understand biomarkers a little bit more. James?
Thank you so much. Thank you, Sabrina. Thank you for having me. It's really exciting. I love being on these webinars. This is such a great forum. So I'll just talk a little bit about the trial, and then we'll sort of have a little bit of talk about the biomarkers in the study. Next slide. So this is, for those of you who've been on the calls, this is our sort of canonical view of the trial. But for people who haven't, the platform trial is designed to be a perpetual adaptive trial where we can make best use of people who are receiving placebo by sharing them across treatment, what we call treatment regimens. And it allows us to randomize more people to receive the active drug. All of the regimens of the study have an open label extension. So when participants or potential participants screen for the study, they then are assigned to what we call a regimen, and each regimen is basically testing one drug or one potential therapy. So they're assigned to regimen A, B, C, or D, and in this case, actually A, B, and C are now enrolled. Once people are assigned to a regimen, they then are randomized within that regimen. And again, because of the way the study is designed, they can be randomized 3 to 1. So for every 3 people that get the active drug, only 1 gets placebo. And then we can share that placebo and get ourselves to a very powerful statistical position for each of the regimens. At the end of the randomized phase, which is about 6 months, all of these regimens have an open label extension where everybody is transitioned to having the active drug, whether they were on placebo before or not. Next slide. So this is designed to be a kind of a late phase trial. That is, these are drugs where we want to know, are they affecting the disease? Are they slowing down the disease? That's our primary question. And we can ask a lot of questions at once in a trial, but that's the primary question. And for that reason, the primary endpoint for all of the regimens so far has been the change in disease severity as measured by our ALS Functional Rating Scale Revised, or ALS-FRS-R. And this is a 12-item questionnaire, and it asks about function in a variety of domains. And it really does hit on some of the very important functions that we ask about even clinically. So it gives sort of a good clinical outcome measure to say, how is the disease progressing? And our goal is to slow the disease progression. Running a trial is a little bit like playing pool in that you need to call your pocket. So you need to decide, if you're going to measure a number of things, you have to decide on one of them to be your primary endpoint. And that's the one that's the primary endpoint here. We also have a number of secondary endpoints, which are traditionally used in trials. So vital capacity, handheld dynamometry is a measure of how strong each individual muscle is, survival. And then number 4, which is what I'll talk about for a few minutes, is treatment-specific biomarkers, and then also exploratory endpoints. Next slide. So one of the things that we thought about when we were sort of designing what we would test in the platform trial is that we had the opportunity not just to look at the primary endpoint and some of the traditional secondary endpoints, but to also evaluate the effect of these regimens or these therapies on biomarkers. I'll explain more in just a minute, and on novel outcomes. So we could actually be building the bridge as we walk over it. That is to say, we have traditional outcome measures, we'd like to do better. We can put some exploratory outcome measures in this trial and biomarkers in this trial, and we can see if they behave even better than our traditional ones. And then maybe we can move ourselves toward having better and better outcome measures over time. So one of the things that we wanted to do is measure DNA. And DNA has many powerful uses that can be used in understanding how drugs are processed in the body. That's called pharmacogenetics. It can be used to look for susceptibility genes. It can be used to look for modifier genes and to balance the populations, or be able to know if there's balance in the populations within a trial. So there are a huge number of uses for DNA. And that has not traditionally been a part of every trial, for sure. We wanted to be sure we included that. Neurofilament is a biomarker. And I should say a biomarker is anything we can really measure quantitatively that tells us about a state of health or disease. And in this case, neurofilaments, we've been exploring for well over a decade in ALS. And we've come to a place where we now understand that by measuring neurofilament levels, this is a protein that we can measure in the blood or in the spinal fluid. By measuring these, we can tell something about the disease. Neurofilaments are proteins that make up the scaffolding of neurons. And when those neurons become ill, the neurofilament proteins are spilled into the blood and the spinal cord, and we can record them there, and they're elevated in ALS. And in people with ALS who have higher elevations in neurofilament, it's possible that they'll have a faster disease progression. We can use this in a number of ways. Number one, we can subset people in the trial and get ourselves more statistical power. And number two, it may be an outcome measure that we could follow. And we know from some recent trials like the tofersen trial in SOD1 ALS that neurofilament can come down and may be treatment responsive. And so we want to test all of those hypotheses and use this to the fullest in this trial. We have other biomarkers that we're testing. We're having we're testing other biomarkers as well. I apologize. It sounds like at least one person's having a hard time hearing. So I'll try to speak up a little. Another biomarker that we're testing in the trial is in urine. And there's a marker called P75, which is a receptor that is expressed on neurons when they develop and also when they become ill. And we can see one of the products of this receptor in urine. And it seems to go up over time in people with ALS. And so we're going to measure that over time and see whether there's any chance that could be an outcome measure in and of itself. And then finally, we're doing some really interesting digital biomarker work. And that is a speech analysis where we're making recordings. And what happens to those recordings is we can actually run them through sort of a automated or even semi-automated kind of analytics pathway. So basically using machine learning to parse it out and say, here's where a person is speaking, here's where they're pausing. When they're speaking, here's the beginning and end of each word. And there are sort of analyses that can do that. And then we can see how rapidly people are speaking. We can look at how many syllables per second they're saying. We can look at changes in the precision of the movements of their tongue or their lips. And we've been able to see that we can actually see changes prior to people themselves being aware of the changes when we use these kind of analytics. But we've never done this in a trial setting in this way before. So that's really exciting. And finally, we're doing home spirometry, which is really the same measure as spirometry that we do in the clinic, but in home spirometry, we're moving that into the home to be more convenient and perhaps to be able to do it more frequently. We have many other ideas, and we have a group that meets every month to look at other ideas of biomarkers that might be relevant that we should apply across the trial. And so that's really exciting and gives us a way to sort of have this be a living part of this trial when that changes as the field changes. Next slide. Just to sort of highlight the progress. Within regimen A, we have 162 individual people who have been randomized. We've met our randomization goal. And within regimen B, it's 167 individuals who've met that randomization goal. And within regimen C, it's 161. So all three of those have now met their enrollment goal. Regimen D is still open and enrolling. And we are rapidly moving toward the enrollment goal there too. And of the people who have been randomized in the trial, 282 have entered the open label extension. That is, they've completed the randomized trial. So this is incredibly exciting news. This is what we sort of imagined when we set out on this journey, and it would not have been possible without all the people on the call, the people who were enrolled in the trial. It really is just amazing to see how the community has come together, and thank you to everybody who's here. Next slide. Sabrina, I don't know if you want to talk a little bit about the expansion.
Yeah, I'm happy to just sort of conclude our weekly update by sharing more news about sort of the expansion of the trial. So we're increasing the number of sites. The sites that you see labeled in blue are part of our first group, 50 sites, and the ones in green are part of our expansion cohort, which is a group of 22 sites that are joining the trial. The two Georgetown University and University of Southern California in the list on the right are the two ones that are already active at this time, and we're working with all the other sites to activate them as soon as possible, and hopefully we'll be able to bring the trial soon to many new geographies or expand capacity where there's many people wanting to enroll. Next slide. And as always, we are joined today by Catherine Small and Alison Bulat, our fantastic patient navigators. They're available to answer your questions by phone or email, and they created a number of QR codes that we are going to showcase today. We have a few new ones to connect directly by using your phone, the camera on your phone, to different websites that they are helping with in terms of eligibility criteria, enrolling sites. And then if we can go to the next slide, there's a few other websites that we thought might be of interest, from clinicaltrials.gov to the National ALS Registry to the NAILS website and the ALS signal from Miami ALS. And why are we sharing these websites? Well, we are because as we continue to enroll in the trial, we also want to, we're very happy to help other trials as well. What we want to do is to test as many drugs as possible, either in the context of the platform trial or in other trials, so that by participating in any of the available clinical trial opportunities that are available at different sites, I think everything will be, everyone's participation will be contributing to finding new treatments. So if you have questions about other trial opportunities, please feel free to contact all these associations. And if you can go to the next slide. We really love this webinar series. As you know, the idea of doing a webinar every week came from our patient advisors, and so you can register for our webinars. You can check the recordings. We're not going to have the webinar next week for obvious reasons. It's it's Thanksgiving, but then we will have 3 more webinars before the end of the year. On December 2nd, we're really happy to have as a guest speaker Neil Thakur from the ALS Association, and then we will have Nicholas Olney from Providence, one of our CPIs, the following week, and we'll conclude the year with a follow-up webinar with Lori Chibnik, our fantastic biostatistician, who gave an initial biostatistical webinar a few weeks ago, and we certainly want her to come back. I know you all asked great questions. Feel free to send them in advance if you want, and she'll be available essentially for a half-hour Q&A. Then we're gonna take a short break, and we'll be back first thing in the new year. Next slide. I think that's where the new code. Today we were talking about how to make sure that we can be present for everyone who wants to have email updates, any updates, news about research about this trial or any trial. Feel free to sign up to the for the ALS Link, which is our weekly, more or less weekly newsletter, where we share the latest information about ALS clinical trials, clinical care, and webinar opportunities. Of course, some of the content is relevant also for our patients who see us at our center, but we really try to always advertise opportunities that are available nationwide, and obviously lots of news about research and other in fact important events that happen around the country. So feel free to sign up. I think you can just point your camera to the screen and it works even during a live webinar, which is still amazing to me. So And with that, I think we can take a few questions, and I'm really glad to have really two of the world's experts on ALS trials today, Dr. Barry, who you just met, and of course, Dr. Cudkowicz, who's with us every week. And so I'll start with a question for you, James, a clarification question about the biomarkers that we will be testing in or measuring in the trial. Can you repeat the name of the urine biomarker?
Yeah, so it's, well, it's got a long name and then we can shorten it. So the long name is urinary P75 extracellular domain. So this is, the receptor is called P75, and P is in PQRS. And P75 is expressed on neuronal membranes. And then it's cleaved off, that sort of the part outside the cell, extracellular, is cleaved off. And that's what we find in urine. So we sometimes just call it P75.
Great. There's another question about the emerging focus on mitochondrial dysfunction and neurodegeneration. Have you considered measuring isoprostanes in urine, plasma, CSF as a standard biomarker for all HEALEY trials?
That is a great question. So just to take a couple of seconds here, mitochondria are the powerhouse of the cell. They're the ones that sort of make the energy for the cell. And if there's a problem in those mitochondria, we can sometimes see basically markers of oxidative stress. And so isoprostanes are a way to measure oxidative stress in the cells. It's a little bit complicated. So the answer is that we are in a few studies measuring isoprostanes. They're very finicky. It happens to be a biomarker that's very finicky. So we're trying to get some experience to see whether we can do this well in a multi-center study. And then I think it's the kind of thing that we could potentially come back to a study like the platform trial, and if it's working well, think about including it. But it has been really tricky to do in a multi-center way because of variability.
Great. So question for you about also relevant for you because you are the co-lead of the CLEAN regimen. There's a question about the recent CLEAN phase 2 trial results. So what are your thoughts on the impact of those results on the likelihood that the HEALEY trial will be positive? Do you think it will, what are your thoughts about that? Yeah, so
first of all, I would say it's a small study. And, oftentimes we're in the situation of having some animal data, some healthy data, data from healthy controls, and then a very small study, and we're trying to read the tea leaves and decide, how likely is it if we go forward in a study. And in a small study like that, the things that we're really looking for are, first of all, we're looking for safety and tolerability. And do we see any problem there? And we certainly did not. We saw that it looked tolerable, it looked safe. The second thing we look for is biomarkers, understanding that many of the biomarkers that we use in phase 2 trials, these smallish trials, are imperfect. And so we're trying to get hints and signals. And I think that there were some trends toward, toward some of the biomarkers looking positive, looking like there could have been a benefit. But understanding this is small and not reaching statistical significance, which is kind of the highest level. And then, we always look at our same efficacy measures like ALS-FRSR, but in a small study, it's very hard to take a signal from that. So I think I feel encouraged that the science that brought this to the platform trial is really exciting. And I think there's nothing in that trial that would make me think that I'm any less excited about it. And maybe there's some signals that boost that excitement, but I have to be really cautious about that. But I'm glad we're doing this study.
I wanted to maybe add to that. One is that I forgot if it was 2 weeks ago or 1 week ago, we had James and Nick, who also leads the CLEAN trial, and the company here. We recorded that webinar. It's on our website to talk more about the data. And then even earlier, we had a webinar about the science behind the CLEAN nanomedicine. So if those are of interest, those are available on the website. And I want to echo that I think the science for that drug is just as good as it was the day before the results from their very small study in Australia came out. So we're still very interested in this drug. And the other thing is that we are testing 2 doses in the platform trial, the same one in the small study from Australia, and then a higher one as well. So it's different that way.
Great. Thank you. Dr. Cudkowicz, I have a couple of logistical questions for you. Can you tell us more about where we are with the analysis of regimens A and B?
Yeah, thank you for asking that. So the analysis of the study doesn't happen until the last person finishes the last visit date. So 6 months from when the last person is enrolled in that arm. So we are talking about next summer for the analysis for A, B, and C. Now that doesn't mean that that we're not cleaning the data, collecting it, and not getting it ready for that analysis. We're doing all that work, and also there is a data safety monitoring board that looks at the data every 12 weeks. For safety as well as for interim analyses. But as far as actually knowing if the drug works or not, that can't happen until the study's actually done, which will be next summer.
Great. And speaking of 2022, can you provide our therapy goal for 2022? How many new arms will be added?
Yeah, that's a great question. I mean, we hope our hope really is to add 2 or 3 at the minimum each year. And so we as you know, are adding regimen E now, and we are in discussions with 4 other companies actually to start, whether they'll all start or not, it isn't quite totally clear. 2 of them we're going to the FDA with. So there's a lot of interest from, companies with very good drugs in joining the platform trial.
Great. And so go back to the biomarker topic with Dr. Barry. Anything new on using imaging to measure neuroinflammation?
That's a great topic and a hot one. So there are a few ways of ways of imaging inflammation in the brain. They use a technology called PET, positron emission tomography, or PET scanning, and we've been sort of refining our understanding of that, sort of those pictures that we get of inflammation in the brain. There are now some early studies that are actually using PET as an outcome measure in their trials. And this is, again, speaking of kind of earlier, smaller trials, trying to understand how a drug works, whether it's getting to where it needs to go to work, and sort of what the chances are that it may be a successful drug in a larger trial. I think there are some really, really powerful uses for PET going forward. So I'm excited to see it being in trials.
Great. Another biomarker question. Are there any new genes identified that may assist with identifying additional biomarkers?
The main focus has been on identifying genes that are causative of ALS. More recently, we have begun to think about genes that may be increased susceptibility to ALS. And then finally, a category of gene that would be kind of a modifier gene. And there's one called UNC13A, UNC13A, which Amar Al-Shalabi has done really excellent, and colleagues have done really excellent work to look at in trial settings to see whether it might influence how people respond to a therapy. I think that's just the start. And the powerful thing about this is that once we have genetics on hand from people in a trial, then we can sort of analyze and reanalyze them. So I'm not sure that I can point to a lot of genes where I say, that gene will be a biomarker, but I think we can point to a couple examples and a bright future for this.
There's a question from someone who is in the trial right now, so a trial participant. Will trial participants get their own personal information or personal data at the end of the trial?
So for biomarkers that are in the trial at this point, the biomarkers that we're including there are a couple of things to say about it. Number one, they're not in clinical use in the way that we think of something like white blood cell count or a hemoglobin count that we measure all the time in people. And so their meaning is still not entirely understood in individuals. And so for that reason, we tend not to be able to kind of share all those results back, although it's something that we are constantly discussing. The other thing, just operationally, is that because these are exploratory biomarkers that we're exploring, we want as little variability as we can have. But the assays are still being developed. And so we do something called batch analysis, which means that we collect the samples throughout the study, we freeze them, and then we run as many of them as we can together to reduce the amount of variability, rather than in sort of a hospital where you might run, you know, one sample at a time, because that's an assay that's been around for 30 years. You know how to standardize it. And I'll just say additionally, in the platform trial, we have the additional kind of complication that we have, you know, we want to plan for the future. So we are not only batching things, but we're also creating pools and being able to bridge today's analysis with tomorrow's analysis. But it means that it's not geared toward giving individual results back.
So there's we answered all the questions that were asked live. However, I do want to acknowledge there were a couple of questions that were sent in before the webinar. So I wanted to take those and maybe ask both of you to answer them and also give us some final thoughts. The first question is about a recent press release about tau research that was done at MGH, and I believe both of you have contributed to that. So the question is, what makes this recent tau research significantly different than the previous studies, and could this lead to new meaningful treatments? So I don't know, James, if you want to start, maybe Merit, you can comment.
Sure. Yeah, it's really an exciting effort led by Gaz Sadri-Vakili here in the lab, who's one of the PhD researchers in our ALS group. And the tau is a protein that is well known in Alzheimer's disease to become misfolded and sticky, and you get these sort of clumps of tau protein that we know are, we'll put it very scientifically, gumming up the works inside cells. And the question was, you know, can we find similar changes in tau and ALS? And previous studies have looked at tau, and they've looked in small samples. And so we had more samples in this study, which I think changes the tenor of the way that we can talk about the results. The other thing is that over recent years, because we've learned a lot about tau from the Alzheimer's field, we can not only look at tau, but look at a modified kind of tau called phosphorylated tau, or p-tau. And we can look at this in a very sophisticated way. And so in the Sadri-Vakili lab, they really looked at a number of even different kinds of p-tau, or modified tau. And they found some really sort of interesting and somewhat surprising results, that overall there was less p-tau in ALS than in controls, but that in C9 ALS there was more. And that may lead us to some interesting biology, because we know that there may be biological subsets. And so I think this is sort of really a clue that we need to look very carefully at this, and potentially could lead to therapies both for ALS and for other neurodegenerative diseases. The last thing is that we looked in tissue, but we also looked in CSF. And there we found this reduced ratio of p-tau to overall tau. So the modified tau was kind of reduced. I think there's as with most stories, it starts out complicated. And as you understand more, you can reduce that to, one sentence or a couple of words and teach it to 8th graders. So we're still at the beginning of this, but I think it could be really powerful.
I don't have anything to add. That was very clear, but I think it was an important paper, and there are a lot of companies working on drugs on that pathway. So there might be some, potential therapies that come from that.
Great. And then the last question that was sent before the meeting, which I think is very relevant for given the topic of tonight's discussion. Has anyone come up with a dependable biomarker to measure the efficacy of a drug, or are we still using the questionnaire known as the ALS FRSR to measure efficacy? James, do you want to start?
Yeah, so neurofilament is probably the closest that we have. It's elevated in people with ALS. It does correlate with the rate of progression. We have at least one study where we saw, the tofersen study where we saw a robust reduction in it. We're still trying to understand how it correlates exactly with the clinical effect. There's been a lot of work on this. It's not specific to ALS, so it's not only the ALS field, so we know a lot about it. We're turning corners with how robust that is. We're still really not quite there, because ultimately the question is this therapy going to make people's lives better and longer? And we need to ask that with kind of clinical outcomes still, but we'd like to get there.
Yeah, I say this is something that's really critical for the ALS field. It will be a game changer when we discover that. It might not be one biomarker, it might be a panel of biomarkers. And it's not surprising that this is a challenging nut to crack, because it is for all brain diseases. It's hard to get the right fluid to do these tests in, but there's been huge progress, and there's, thanks to many of you, large repositories of samples from people with ALS and clinical data. And I think this is a ripe opportunity for funding agencies, for example, to really go on a big campaign to fund companies and academics to use those samples and try to make some new discoveries. And last thing I'd just say is that is one of the pluses of the platform trial is other platform trials in oncology and other fields have developed these surrogate biomarkers because they have these bank samples, they have the clinical data, and they have the possibility to keep learning from it. So we're hoping that we'll be contributing to that through the platform trial as well.
Excellent. Thank you. And I know somebody asked about how can someone be considered for regimen E, and I think the answer is always to connect with Catherine and Alison, our patient navigators, who can help identify sites that are close to you. And Merit or James, any final word?
I just want to thank you for inviting me, but I also thank people for joining. This is such an amazing community, and I just love the times that I get to be involved.
Yeah, I'll add my thanks, and thank you for helping us finish enrollment for the first 3 and almost done with the 4th. It's really quite amazing and gets us just closer to getting the answers for these initial drugs.
Thank you. Happy Thanksgiving, and we'll see you the week after.
Thanks,
Thank you.
Thank you.
Bye-bye.
Goodbye.